direct observation of evolution is possible. Now, one thing it's been very difficult for us to observe because we think of evolution happening over very long periods of time is the evolution of the larger lineages of organisms, kingdoms, phyla. Um, but what's very much simpler for us to see, because it's happening within our time frame, is microevolution, which isn't to say that macroevolution, or the evolution of new species, genera, families, etc., there's a lot of strong evidence for that, but microevolution is a lot easier for us to see directly and to empirically measure and calculate. Microevolution is essentially changes in the genetic makeup of a population over generations. So here we're going to start talking about genes and alleles again, and 5 agents of microevolution and some examples of these 5 different agents. So here we have to talk about the Hardy-Weinberg equation. So let's assume that we've got a gene in a population, and that gene has 2 alleles, and we'll call them A and little a, big A and little a. So just like a simple dominant recessive, 2 alleles, just like with Mendel's P's, which means that we've got 3 genotypes, big A, big A, big A, little a, and little a, little a. Okay, so we can calculate the frequencies, or really the percentages of the alleles and the genotypes. So we'll call the frequency, or the percentage of the total number of alleles in that population as big A as P. We're going to call the frequency, or the percentage of little a alleles in the population as Q. So what this means is that for all the alleles, we can sum up these allele frequencies and say that the percentage of big A alleles plus the percentage of little a alleles is going to equal 1, or 100%. So all of the P's that Mendel grew are 100%, and each of them had either a big A or a little a allele, and we can say, well, what were the proportions? Was it 60% big P and 40% little p? Possibly. But whatever it is, it's going to sum up to 100% of all of the alleles. And then for the genotypes, we can say that P squared plus 2PQ plus Q squared equals 1. Or big A, homozygous dominant, and little a, that Q squared term. And then remember, from a Punnett square, you end up through those Punnett squares with 2 boxes that are big A, little a. So that's why we have 2 times PQ. And that's going to sum to 100%. And if you are lost in all of the math here, you might be asking, why math? Why now? We've gotten to this point in the semester without very much math at all, and now all of a sudden we're getting these not only math, but math involving the alphabet, the worst kind of math, in my humble opinion. And the answer is we're not going to get too far into it. But the reason I bring this up is because this Hardy-Weinberg equation is a way that we can model allele and genotype frequencies through time. And a couple of very important things about it is that it demonstrates that we can measure and quantify these particular aspects of change through time in biological systems. In other words, we can measure evolution and demonstrate that it's happening. And what Hardy and Weinberg came up with in their equations was they realized that if you obeyed these 5 simple rules, then you would not see any change in allele frequencies or in genotype frequencies. So in order to make sure that from one generation to the next, none of those numbers would change, these 5 conditions had to be met. One was no natural selection for those genes. It couldn't be that big A or little a was any better at helping those organisms to survive and reproduce. Number two, random mating. Nobody could select a partner to mate with. So there's no, you couldn't choose that if you were a purple flower that you would only cross with purple flowers. Or if you're a white flower, you only cross with white flowers. No sexual selection at all. Had to be completely random. Rule 3, no immigration or emigration. Nobody could come in. Nobody could leave. You couldn't add any, because that would throw off the numbers. That would throw off the frequencies. Just some schmo off the street, some schmo flower comes in. It's going to change your numbers. Fourth condition is that really, to make it work, you had to have an infinitely large population size, but that's not mathematically possible. So we'll just say you have to have a very, very large population size in order to make this work. And that doesn't always happen. And the fifth is that you couldn't have any new alleles. You couldn't have a new allele for this gene, or that's going to throw off your numbers, because you're not going to have 100% of all the alleles anymore. If you just have, you had big A and you had little a, and all of a sudden you've got alpha or something else, some other allele for that gene in the equation, of course things are going to change. So one way to look at Hardy-Weinberg is to say it's what scientists call a null model. It says this is what we would see if there were no microevolution. What that means is if any of those 5 things is happening, there's going to be a change in allele frequency and in genotype frequency. In other words, when those 5 things happen, any of them happen, you're going to get microevolution. So basically, take those 5 assumptions of the Hardy-Weinberg equilibrium that says that there's not going to be any allele change or any genotypic change. And if any of them flip, and you go from not having natural selection to having natural selection, things are going to change. So another way to say that is things that are going to lead to microevolution are natural selection. Maybe big A is individuals survive and reproduce better than individuals with the little a allele. Non-random mating. If there's sexual selection, if you get to choose who you're going to have offspring with, that is going to change allele frequencies. Immigration and emigration, otherwise known as gene flow. Let any random schmo off the flower boat in, and that is going to affect allele frequencies. If you limit population size, if you just have a random sample of a population, then chances are you're not going to have all of the alleles represented equally. Just like if you reach your hand into a bowl of M&Ms, chances are you're not going to get an equal number of M&Ms of each color. You're going to get a random sample. So maybe you're going to get a whole bunch of browns and a couple of yellows and a red and a blue, but maybe you're not going to get any green ones. So that's an example of what happens when we limit population size. And then the last one is if we allow mutations to occur, that's going to generate new alleles, which is going to definitely change allele frequencies. Okay, so natural selection, as I mentioned previously, is the idea that through generations, certain heritable traits that make organisms better matched to their environment are going to get passed on to the progeny, and over time, they will be more abundant in the population. And what we have in this picture here are 3 different modes of natural selection. The first one, the one on the top there, is called disruptive selection. So say we've got a population of these scallops here, and they range in color. They have a heritable range in color from sort of grayish to more brownish. And at the beginning, before there's some selective pressure that's applied, the majority of the individuals are sort of this in-between color. And then say something happens and the geology of the beach shifts. And there's a lot of grayer rocks and a lot of browner rocks. And sort of tanner rocks in the middle aren't as good a hiding place for these scallops. So what's going to happen is over time, it's more adaptive to have either one appearance or the other. So over time, after this disruptive selection event, you're not going to have as many of these tan-colored scallops. You're going to have much more of the gray and the brighter sort of, I don't know what you call that color, scallops. Another type of selection or mode of selection is stabilizing selection. So I think of stabilizing selection kind of like being extra medium. So here we've got a population of lizards. We've got some that are sort of shorter in their total length and squatter, and some that are longer and skinnier. And then between we've got an in-between condition where they're not too long, but they're not too fat. So there could be a stabilizing selection pressure that says, well, if you're too skinny and thin and long, then you're going to be selected against. Maybe you can't. You're not as sturdy. You can't climb up trees as fast, or you're easier to catch because you can't run as fast because your legs don't have as strong of a musculature. But then maybe if you're too short and too squat, maybe it disables you in some other way. So stabilizing selection says over time there's going to be sort of the middle state, and there's going to be more individuals of this middle state because that is what is best selected for. And there's a disadvantage to being either too much in one direction or the other direction. That's stabilizing selection. Directional selection is a third mode of natural selection. And in directional selection, it says, well, it's not going in both directions like we see in disruptive selection simultaneously. It's not selecting for the middle state. It's kind of pushing towards everyone in that population towards one direction. So, for example, in this bottom state here, we have different, looks like drafts, that maybe in the beginning of this exercise, they had fairly short necks, they were fairly short individuals, but over time, maybe there were fewer and fewer leaves that could be accessed by organisms that were shorter. And so not by stretching their necks, but by having only the longer-necked individuals surviving and reproducing, being fit, over time, you're going to have organisms that have very, very long necks that are very tall. So that's directional selection. Sexual selection means basically non-random mating. And it means that you pick a mate, and there is a lot of evidence for sexual selection out there. There are a lot of organisms out there, like this peacock spider, doing this crazy dance that the only reason that a peacock spider looks like this, as far as any scientist can ascertain, is to attract a female peacock spider. Female peacock spiders don't look like this. They don't have these elaborate dances. They don't have these very brightly colored abdomens, but it's all for showing off to try and reproduce with a female. And Hardy Weinberg says if you get to pick your mate, you're not going to have allele frequencies and genotypic frequencies be stable, and you're going to see microevolution. Gene flow means you're going to have immigration or emigration. You're going to have movement of alleles or genes. Just like when I was talking about the dogs and why is it that we say that dogs are a single species, even though Chihuahuas and Great Danes don't make great parental couples. It's because there can be gene flow. There can be organisms that harbor genes from all of the different representatives of that species, even if they can't mate with each other directly. So immigration and emigration are kind of part of the notion of gene flow, but it's kind of a bit of a bigger concept when we think about how different organisms are kind of vessels for the different genes. So in this population over here on the left, we get some immigration, and this brownish beetle is going to introduce brownish heritable genes into this population of green beetles. Genetic drift, or limited population size, it's kind of a sampling artifact, kind of like the M&M example. And the classic example of genetic drift, or the effect of a small population size that's not due to selection, is called the bottleneck effect. And what happens in the bottleneck effect is, imagine, you can tell from the picture, if you've never seen a bottle before, this is what it looks like. And we have a big population of marbles in the bottle. And now let's say we want to take some marbles with us to show our friend somewhere else our marble collection. We can't take all our marbles. We can only take a small sample of those. So as we pour them out of the bottle, all the marbles are the same shape, same size, but they're different colors. So what we get in the cup isn't necessarily going to be a representative sample from the bottle. And if you look at the picture, you can see that none of those red marbles are making it into the sample that we've taken. So this sampling artifact is called genetic drift. It's the effect of limited population size. Finally, mutations. And again, mutations in a biological sense means changes in the DNA, changes in the genes. New alleles can be formed that make new proteins. And if these are in the gametes, they can be passed on to the offspring. If mutations are present in the gametes, that means it's a heritable variation that can be passed on to the progeny. So here, for example, is our original DNA codon, TTC, which at the mRNA level makes the complementary AAG, which is going to encode lysine. But let's say instead of TTC, we've got TTT, and that changes our messenger RNA codon to AAA instead of AAG. But because of redundancy in the genetic code, we're still going to encode lysine. So that's a silent mutation. Well, what instead of changing TTC to TTT, we change it to ATC? Whoa, then at the messenger RNA level, the codon becomes UAG. And UAG is a stop codon. And that's what we call a nonsense mutation. Because we've changed an amino acid codon to a stop codon. And that means that instead of adding in lysine, we're just going to stop making the protein altogether. And nonsense mutations typically are pretty catastrophic because it means you are likely to lose the function of that protein. And then, what if instead of changing that first T to an A, we change the middle letter from a T to a C? So our DNA codon is TCC, which makes our messenger RNA codon AGG. We've changed from encoding for lysine to arginine. Fortunately, both lysine and arginine are basic amino acids. And so maybe that's not going to have much of an effect on the final protein product. But what if we change that to TGC, which is going to encode a CG, which means we're going to encode threonine instead of a basic amino acid. Now we've got a different type of amino acid, a polar amino acid. That's probably going to change the protein. It's going to change the final protein product because of the chemistry of that different side chain. So these are all different types of mutations. And when you introduce a mutation, you're basically introducing a new allele into the population, and that's going to lead to microevolution. So these are, again, our 5 learning objectives from Chapter 27. And we're going to end up with our final lecture on some aspects of ecology.